Good afternoon, everyone. Hello, my name is Barbara Cannon, and I'm the Chief Operating Officer of NuSirt BioPharma. And I apologize for the brief delay. We're just trying to get our audio all set up for you. As you know, please keep the speakers on mute until we have the opportunity for the Q&A session, as this entire presentation is being recorded. Again, it's a pleasure for me to welcome you to our virtual investigator meeting of NSWM01. I'd like to start off by introducing a couple key contributors to the protocol, our KOLs, Dr. Frank Greenway and Dr. Stephen Smith. Frank Greenway is an endocrinologist and directs outpatient research at the Pennington Biomedical Research Center. This center is focused on obesity and located on the campus of Louisiana State University in Baton Rouge, Louisiana. Dr. Greenway is on the editorial board of the International Journal of Obesity and has published over 150 peer-reviewed articles, over 30 book chapters, predominantly in the field of obesity, and holds several patents. His interest is in developing new treatments for obesity and has been active in clinical trials of candidate pharmaceuticals, dietary herbal supplements, medical foods, and devices. Pardon me. Stephen Smith is also an endocrinologist. His work bridges the gap between molecular biology and clinical care. Based in Tampa, Florida, at the Translational Research Institute for Metabolism and Diabetes, his work involves translating discoveries in the basic sciences into clinic and using clinical knowledge to direct the basic scientist. Dr. Smith's research is focused on obesity, diabetes, and the metabolic origins of cardiovascular disease. He is specifically focused on how individuals differ in their ability to adapt to diets high in fat and understanding how obesity leads to type 2 diabetes. Dr. Smith has published over more than 160 peer-reviewed scientific manuscripts, reviews, and/or chapters, and is active in medical education. Turning to our NuSirt colleagues, I'm pleased to introduce a valuable member of our scientific advisory board and your medical monitor for this study, Dr. Orville Coulterman. Orville serves as adjunct professor at the University of California, San Diego Medical Center in the Division of Endocrinology and Metabolism. His prior work at Amylin Pharmaceuticals includes a wide contribution serving in numerous capacities as medical director, senior vice president of clinical and regulatory affairs, as well as senior vice president of research and development. Dr. Coulterman has authored numerous research publications and is a recipient of several honors and awards from the American Diabetes Association. He is currently serving as the principal investigator of the EPIC study, that's Epidemiology of Diabetes and Control, at UC San Diego. Most importantly, should you have any medical or clinical study questions, I know you will find Orville to be extremely knowledgeable, approachable, and a pleasure to interact with. Next up is another of our colleagues, Dr. Michael Zimmel, who you will be hearing from momentarily. Michael is NuSirt's founder and chief scientific officer. Michael started NuSirt in 2007, leaving the University of Tennessee to join the company full-time in 2012. He is a recognized expert in energy metabolism and endocrine, pharmacological, and nutritional modulation of chronic disease risk. He is the author of over 200 peer-reviewed publications, primarily in the regulation of fat cell metabolism and application of this research in the prevention and treatment of diabetes, obesity, and metabolic disease. Michael and NuSirt holds an impressive intellectual property estate with a number of patents and regulation of energy metabolism and the coverage of new therapeutics for diabetes, obesity, and metabolic disease. Lastly, let me introduce Dr. Omar Flores. Omar has been your face of NuSirt BioPharma, and he's responsible for the overall conduct of this obesity study. Dr. Flores holds a PhD in molecular and cellular pharmacology from the University of Miami and an MBA from the University of North Carolina, Chapel Hill. Omar brings a unique intersection of science and business. He was a Hoffman-LaRoche Fellow at Duke University, investigating the role of small RNAs in viral pathogenesis, and has authored 8 peer-reviewed publications and a recipient of an NIH predoctoral fellowship. I met Omar when he was recruited to Mountain Group Partners as a financial analyst. Mountain Group Partners is our lead investor group for NuSirt BioPharma. And as I was setting up the clinical operations for NuSirt, I quickly identified, or as my colleagues would say, I marked Omar's skill sets and eventually lured him from venture capital to R&D operations. You will find Omar to be very responsive, astute, and dedicated to make this clinical study a success for all parties. He will be visiting you again soon in 2018 and looks forward to sampling your local cuisine. So with that, I will turn this meeting over to Michael. Michael?
So this is Michael Zemmel, and hopefully you can see my slides. I'm going to go through them much faster than I had anticipated. Some of you may have seen a few of them when I thought I was being live audio, but apparently was not. So I'm going to just give you some high points. Our platform targets the enzymes that are nutrient sensors. They sense caloric restriction, and we activate these to recapitulate the benefits of caloric restriction, including reduced lipid storage and increased lipid oxidation. Our basic technology is built around the amino acid leucine, which enables the coactivation of SIRT1 by other activators, both SIRT1 and AMPK by other activators, such as metformin. And the result is a profound synergy, and we've demonstrated that clinically. Again, I'm zipping through these. Our integrated mechanism of action for the compounds that are in NS-WM-01 is shown in this slide, and that is leucine plus co-activating compounds recapitulate the effects of caloric restriction, recapitulate the effects of exercise, and oppose the effects of caloric excess. They do this by activating SIRT1, AMPK, and off on the right you'll see eNOS, endothelial nitric oxide synthase, which converges on and also co-activates SIRT1. The net effect is reduction in weight, but also improvement in multiple obesity-related comorbidities. We have a vasodilatory effect resulting in a drop in blood pressure. We improve insulin sensitivity. We reduce circulating lipids. Now, specifically in this trial, we are going to be testing a combination of leucine and very low-dose sildenafil, and a combination of leucine, metformin, and very low-dose sildenafil. The leucine and metformin activate both SIRT1 and AMPK, mimicking the effects of energy restriction. Sildenafil you normally think of as a PDE5 inhibitor, and obviously it does that, but at much lower concentrations, it's also an activator of the eNOS, or endothelial nitric oxide synthase system, leading to an increase in nitric oxide, which further activates SIRT1, or further recapitulates the benefits of caloric restriction. So that is the framework for what we're doing. Now the question is, how does it really work clinically? Well, overall, data to date show that activation of this pathway through our obesity platform results in a suppression of fat synthesis in fat tissue, coupled with an increase in fat utilization, fat oxidation, as well as mitochondrial biogenesis, or making new mitochondria, in skeletal muscle. So the net effect is transferring energy from storage in adipose tissue to burning in skeletal muscle. So to give you a sense of data to date, we ran a 16-week clinical trial, NS-0200-01, in which we compared 2 doses of NS-0200. NS-0200 is a combination of leucine, metformin, and low-dose sildenafil. And here we compared, we've already optimized the leucine and the metformin. Here we are comparing the effects of very low-dose sildenafil, 0.5 milligram, low-dose sildenafil, 1 milligram, in combination with leucine and metformin, and both, of course, compared to placebo. And what you see on this slide is a nice linear weight loss over 16 weeks in all subjects who completed the trial and adhered to the protocol. If we look at subgroups, you see comparable. Here we have individuals with elevated liver enzymes. So these were non-alcoholic fatty liver disease patients who were a little sicker, and you see that they had even a greater weight loss over 16 weeks. Again, it's fairly linear, no indication of a plateau. It's not just weight, it's also obesity-related comorbidities. So here we're showing you liver fat as an obesity comorbidity, and what you can see on the left-hand side is the absolute value, and on the right-hand side, the percent change. And what you can see is the high dose of NSO 200 caused about a 15% decrease in liver fat. Now, if we look at another slice of patients, here we're looking at patients that had elevated triglycerides, that is, using the standard clinical cutoff of 200, people who had triglycerides greater than 200 milligrams per deciliter. And what you see is, again, a somewhat linear weight loss with overall about a 5 kilogram weight loss over the course of the trial. This was accompanied by, as you see on the next slide, a very nice drop in serum triglycerides, nearly 150 milligram per deciliter drop in serum triglycerides with both doses of NSO 200. There was a tendency towards dose response, but really the two doses were equivalent to one another. Likewise, if we look at blood pressure, this particular slide, it shows obese subjects who were on antihypertensive meds at the time of the trial. But we get a comparable effect if we look at the full cohort. We get a comparable effect if we look at individuals who were hypertensive regardless of whether they were on meds or not. In fact, if we look at that group, we also show a very nice drop in systolic pressure. Here you see a nice drop in diastolic pressure. If you look at hypertensives regardless of medication, we get drops in both. Now, I'm going to turn to a little bit of preclinical data to help you understand why we're studying leucine sildenafil as well as leucine metformin sildenafil. We have studied this in a couple of trials preclinically, and in one trial, leucine sildenafil actually looks better than leucine metformin sildenafil, but in a recently completed, more comprehensive trial, what we see is that with very low dose sildenafil, the leucine sildenafil on the in the left-hand panel seems to outperform leucine metformin sildenafil. Here you're looking at weight loss in obese mice over 12 weeks. The black line is control, the blue line is leucine metformin sildenafil, and the purple, or whatever color that is, line is leucine sildenafil. If you look, however, at the right-hand panel, what you see is that when we boost the level of sildenafil up to a human equivalent of 4 milligrams, there we see that leucine metformin sildenafil looks equivalent to leucine sildenafil. Since we already have good clinical data on the triplet, we feel obligated to study both the doublet and the triplet in this study in this upcoming study. So just to give you a quick sense of how we've chosen our doses, again, we have spent a lot of time optimizing our leucine and metformin doses. Leucine we've optimized based on plasma leucine levels to get to a level that is sufficient to activate SIRT1 without any untoward effects. Metformin that is based on optimizing for insulin sensitivity based on work from a previous clinical trial. And for sildenafil, we are looking to activate endothelial nitric oxide synthase without inhibiting PDE5. And once you get north of about 5 milligrams, you begin to get some inhibition of PDE5. So we're trying to keep our dosage below that. So I've moved through this as quickly as I can, given our technical issues. Here's a quick summary. We know that leucine and sildenafil, with or without metformin, activates the signaling pathway that mimics caloric restriction. Clinically, we know that the triplet of leucine, metformin, sildenafil induces weight loss and improves obesity comorbidities without any caloric restriction. We have preclinical data to suggest that leucine sildenafil may be better than the triplet combination, but we don't have clinical data available on leucine sildenafil without metformin. And as a consequence, we are conducting this study to compare the doublet versus the triplet combination, as well as to optimize the dose of sildenafil used in either the doublet or the triplet. With that, I want to make sure we stay on task and on time given our long delay, but I can take a question or two if there are any. If not, we will progress.
Yeah. Okay, we have here, through many technological devices here, Dr. Orville Coulterman to go through the inclusion and exclusion criteria. Orville, can you go over the protocol highlights and the inclusion and exclusion criteria?
Sure. Need to collect my thoughts after all this, concept. But basically, it's a parallel study, with 4 arms, looking to evaluate 2 combinations of leucine and sildenafil, 2 different doses of sildenafil, and then the same 2 arms repeated with the addition of a gram a day of metformin, given as 500 milligrams in the morning and 500 milligrams, in the evening. The primary endpoint is the mean % weight loss for each group, and then there are a number of secondary endpoints. In terms of the inclusion, exclusion criteria, subjects need to be over the age of 18, under the age of 65, or 65 or less at study entry. If female, they need to not be breastfeeding. They need to be postmenopausal or have a negative serum, pregnancy test, and then if they are childbearing potential, which include perimenopausal women who've had a menstrual period within the past year, you must be willing to practice appropriate birth control using a method that has a documented low failure rate. And the details of that are outlined in the protocol. Subjects should have a stable body weight, defined as plus or minus 5%, and no change in their health over the last 3 months. And this is based on medical history alone. Their BMI needs to be between 30 and 45. They should have a stable diet for the past 3 months, and no acknowledged plans to undertake drastic dietary modifications. If subjects are using antidepressants, excluding those that are called out in the exclusion criteria, they should have been on a stable dose for at least the past 3 months, and project to be capable of maintaining the stable dose throughout the duration of the study. The clinical laboratory test, hematology, chemistry, and urinalysis need to be within the normal range, or not, clinically significant, based on the investigator's judgment, and or the abnormalities that are known to be associated, with obesity. Now, as we go through the study, if there are questions about whether something is clinically significant or not, please get in contact with me so we can discuss. And then finally, they need to be capable of reading, understanding, and providing informed consent by signing the informed consent document, and then also the appropriate HIPAA form. moving on to exclusion criteria, subjects cannot have a diagnosis of diabetes or be using any anti-diabetes medication to participate in the study. Patients are excluded if they have severe renal impairment defined as a eGFR less than 30 mg per milliliter per 1.73 meters squared of body surface area. Patients with chronic metabolic acidosis are also excluded, and that's defined as calculated greater than 25 millimole per liter without including potassium, and normal values are 8 to 16, but acidosis is defined in the lab we're using as being greater than 25. And there's a list of medications which the subjects cannot have used during the preceding 8 weeks prior to entering screening. This includes the anti-diabetes medications, including metformin and any combination drug that contains metformin. I think the reasons for that are obvious. And for the same reason, sildenafil, tadalafil, or vardenafil are excluded, as are OCT2 and MATE inhibitors, which is cimetidine, quinidine, pyrimethamine, glycocyclase stimulants are excluded, alpha blockers, nitrates, potent CYP3A4 inhibitors, and there's a list of those included in the protocol. All antihypertensive medications, since we are looking at blood pressure as one of the outcomes, and then medications associated with, or known to be associated with, weight changes. These include drugs approved for the treatment of obesity, cyproheptadine, medroxyprogesterone, the atypical antipsychotic drugs, tricyclic antidepressants, lithium, MAOs, glucocorticoids, SSRIs, and or SNRIs, the analeptic drugs, systemic corticosteroids, and stimulants such as amphetamines. Also, finally, any dietary supplement that is labeled for the purposes of weight management or maintenance of healthy weight will be an exclusion. Diagnosis or evidence of eating disorders, and in alignment with the inclusion criteria, a weight change greater than equal to 5% of body weight during the past 3 months. Also excluded are subjects who've had bariatric surgery, subjects who've had an infection requiring antibiotic treatment within the last 30 days, any chronic inflammatory disease such as inflammatory bowel disease, rheumatoid arthritis, other autoimmune entities, diseases interfering with metabolism, inborn errors of metabolism, myxedema, Cushing's disease, maple syrup urine disease, etc. They also are listed in the protocol. A history of alcohol abuse, which is defined greater than or equal to 21 drinks per week of alcohol for males, and greater than 14 drinks per week for females. So that's within the last 3 months. So history of heavy alcohol use in the distant past would be permitted, and then subjects that fail a urinary drug screen. History of substance abuse, opioids, which would probably be a big one in this day and age, and then marijuana is also included. any subjects that's received an investigational drug within 3 months of screening, and then the usual designation that subjects who have donated blood within 3 months or plan to or see a need for donating blood during the projected duration of the study. Any other medical condition that leads to a life expectancy of less than 2 years. This includes known cancers. Patients that have been diagnosed with metastatic carcinoma in the last 5 years are excluded. Subjects that are known to have allergy or hypersensitivity to metformin, sildenafil, or leucine. And then blood pressure criteria: resting hypotension, defined as a blood pressure systolic less than 90, a diastolic 50, or a diastolic less than 50. And then hypertension defined as a systolic greater than 170, diastolic greater than 110. Cardiac failure, coronary artery disease, including unstable angina, what are exclusions? History of congestive heart failure, evidence of left ventricular outflow, obstructions, aortic stenosis, subaortic stenosis due to hypertrophy, and then any significant abnormality of the autonomic control of blood pressure. Even though the doses of sildenafil are low, there's a need to exclude males who are at risk for priapism due to anatomical deformities or other predisposing conditions such as sickle cell anemia, multiple myeloma, and leukemia. Clinical evidence of hepatic impairment or ALT or AST values that are greater than 5 times the upper limits of normal. And then finally, the usual exclusions of immediate family members, personnel directly affiliated with the study, or subjects that are employed by NuSirt BioPharma.
Exclusion.
Okay, so any questions about that?
Hi, this is Dr. Mohseni from Kalina Research. I do have a question regarding for subjects who have the medical marijuana use. Since you're allowing prescription meds for opioids, are you allowing someone who has a license for medical marijuana?
That is a California issue these days, isn't it? I would prefer to exclude those patients.
You prefer to exclude those patients?
Yes.
Does medical marijuana make you hungry like smoking marijuana for recreational purposes?
It's reported to in some cases.
Yeah.
And the real issue that we have is that, at least in California, there's a blurry boundary between medical marijuana and recreational marijuana use.
Okay. So, all right. And I also have a question regarding exclusion number 3, where you're defining acidosis as greater than 25 millimoles. Is that for the anion gap? Is that what we're calculating?
Yes, that's what we're referring to.
I'm sorry, I couldn't hear you.
No, that is what we're referring to as the anion gap.
Anion gap. Okay, great. Thank you.
Any other questions for Orville on the inclusion-exclusion?
Hi, Orville. This is Mary Kaczynski at Premier. I had a question about some of these that are listed in K. Is there any kind of washout period, or is that washout period going to be the 3 months that you referenced before?
So for the ones that are listed, there, think if we, as a general rule, if the patient's been off for, more than a month, but, if you run into questions with one of those agents, let's talk about it, and I will, look at the, pharmacokinetics of those drugs and make a judgment as to whether we can let a patient in. But certainly, if the patient has been off of these drugs for, greater than a month, they would be allowed in.
Orville, so currently we have it written so that 8 weeks prior, we want them off, but it sounds like if they have a question, they can contact you and you can have a discussion with them on it.
Correct.
Antibiotics as well. That all the antibiotics. Anything within the last 30 days. So even if they had an ear infection or sore throat or anything.
Yeah. So let me have an awkward conversation here with my colleagues. I don't recall why we included antibiotic exclusion. So Omar, can you help me?
Yes, so we're talking about, I believe it's exclusion number, give me 1 second and I will find it.
It's 4A.
Yes. So anti-diabetes medications, is that what you're talking about? Antibiotics?
Oh,
or?
I'm sorry. I have the wrong one. No, it is
Yes.
number 8.
Yes, yes, an infection requiring antibiotic treatment within the last
Yes.
30 days. So basically any major
Yes.
infection.
Yeah,
So,
we're just curious because we are coming into the cold and flu, sinusitis, those kind of infections. So we're just curious about that.
So I think we were intending for this to be related to really significant infections. So as I read this, as I was going through it, I flashed the bronchitis that you're referring to, Mary, as well as routine urinary tract infections.
Okay.
So if you have patients that are impacted, let's talk about those and as well and handle them on an individual by individual basis.
Okay. And then the only other question we had here, if I could just ask one more, is if we have a patient who is below the HbA1c of 6.5, but we see over the course of the study that increases, should we just notify you, or you guys have to be watching that as well? Because we'll probably want to treat that or do something about that.
Yeah, so we need to handle those on a case by case basis.
Okay, sounds good. Thank you.
Is the BMI between 30 and 45, is that inclusive?
Yeah. So it's over 30, and it can be less than or equal to 45.
Less than or equal to 45, and it has to be over 30? Okay. It can't be 30.
It can be 30 the way that we've written it, Orville. It can be 30. 30 is inclusive.
Inclusive,
Sure.
so it is inclusive. Okay.
Yes.
Just want to be sure about that.
Yes.
Any other questions for Orville? If not, we'll move on back to Michael, who will just quickly review the waist circumference and blood pressure measurements.
Okay, so I'm going to make this quick because without... Yes, I'm going to make this quick because without video, it's going to be a little silly for me to do anything but quick. The key thing is there is waist. I want to talk to you about waist circumference and blood pressure because these are really important secondary outcomes to us. And so you already know you've been provided with measuring tapes for waist circumference. The key here is to make sure you have the anatomical marks for waist circumference. So you're going to go to the midpoint between the iliac crest and the lowest rib that you can feel, and find the midpoint. You can mark it. The key for using the tape, there's really only two keys. One is to make sure it is parallel to the floor. The position of the patient is already shown in your protocol, that is standing with their legs apart. The key is to make sure that you are parallel to the ground and that you put enough tension and not too much tension on the tape. And so we provided you with these nice tensioning tapes, and as you pull it taut, I don't know if any of you are playing with these in your hands while I'm talking to you or not.
They have not received them yet, Dr. Zimmel. They will receive them in 2 weeks.
My mistake. Well,
You'll have to describe
Tantalize them with it, yeah.
so basically, at the end of the measuring tape, there is a spring-loaded device that is about 2 inches, inch and a half in length. And as you pull it taut, there are 2 orange balls that appear, separated by a silver disc. And what you want to do is pull it taut enough to show the first orange ball and show the silver disc without seeing the second orange ball. If you're not holding it in your hands, it's not going to make sense, but you will read the instructions and they will be as clear as daylight. I'm so sorry you don't have that in front of you, but it's very important that you apply a uniform amount of tension, and these tensioners are designed for that. The second important issue, again, is to make sure you have the appropriate anatomical landmarks and go between them. Now, with regard to blood pressure, and Omar's going to switch the slides for me, I think. So, with regard to blood pressure, again, another important outcome, and you will be receiving or have received them?
Will be receiving.
You will be receiving these blood pressure monitors. Included in the blood pressure monitor set will be several cuffs. First thing that I think is important to mention is that you select the right cuff size. Each cuff has an index and a range, and as you wrap the cuff around the arm, the index needs to be within the white bar of the range. Again, this will be obvious when you're holding cuff in hand. If the cuff is either too big or too small, you can go to the next size up or the next size down. You will have appropriate cuffs to cover all possible subjects. Second thing is, these are automated, unattended blood pressure measurements. So you want your subject, you want your subject seated with arm supported, non-dominant arm supported. It can be on the edge of a chair, so the arm is approximately at the level of the heart at the time the blood pressure is being measured. Once you set it up, you will set the machine up. Once you have the individual settled in their chair with the cuff on, the machine will be set to take 3 measurements at 2-minute intervals apart.
Yes, and you will have to set that part up. And it's very important for you to pay note to Dr. Zimmel saying that there's a 5-minute delay before the first measurement, but then the intervals between the first and second, and the second and third are 2 minutes apart, because we did not write that in the protocol, which we should have. So that is one note that you will have to make, and you will set all of these up.
Correct. So these will, yes, you will have to set these up. The instructions that come with the monitor are quite clear in doing so. But it is important that these be unattended blood pressure monitor measurements. We want to make sure that we don't get the white coat effect, which is often very pronounced, and we really need to have uniform blood pressures. Again, without the monitor in front of you, that's about all I can say, and in the interest of time, I'll move on.
All right. So we are at the last part of this, and I will quickly go through the visit schedule. This is a very straightforward visit schedule. Give me 1 second while I pull it up. As you can see, you have visit 1, which should be about a week before the baseline visit. The baseline visit will be the base from which all of the other visits are determined. So they're not determined from the last visit. They're all determined from baseline. As you can see from this particular schematic, every visit is 1 month apart, and you have a phone contact in between each one of these visits. All of the different tests that you will be running and different items that you will be discussing are listed here at the back of the protocol. And if you have any questions on this, I'd be more than happy to answer them. There's really nothing specific on here that we needed to go over. The phone contacts, you'll be discussing adverse events and going over medication compliance. You will be collecting labs at visit number 1, screening obviously at baseline, and at visit 5 and at visit 8. So every 3 months you're collecting labs after randomization. And really, this is one of the more straightforward protocols that we've ever had. So there's really nothing in here that's tricky or that we really need to explain any further. So I'd be more than happy to take any questions on this visit schedule.
Omar,
Yes.
this is Rachel from Dr. Jennings' side in San Antonio. On section 4.5, the third bullet, it states a 10-hour fast, but then in section 6, it states an 8-hour fast.
Yes, ma'am. It should be a 10-hour fast. We apologize. We had thought we had caught every one of those that said 8 hours instead of 10 hours. The ICF states 10 hours, and we fixed the ICF correctly, but I did miss that 8-hour one. It is the 10-hour fast.
And then do you know how many blood pressure machines the sites are going to be receiving?
Yes, you will receive 1 blood pressure machine, and you will receive 2 waist circumference tape measures.
Okay, thank you.
Hey, Omar, this is Mary from Premier. We have one question for you. Patients that are on other medications and they come in for fasting, are they allowed to take those medications in the morning?
I will let Orville, I believe he's still on the line, answer that question. I believe if it's with water, that should be fine, but I will let Orville answer.
So if it's a medication that is time critical, they obviously should take it, but take it with water. If their meds are not time critical, it would be best if they could come and bring their meds with them and take the meds after the blood sample is drawn. But certainly for medications that are time critical, they should take them prior to the visit, prior to coming in, so that they maintain their schedule.
Okay. And then we will have them dosed on site after we draw their labs, or?
Yes, that first dose will be taken on site. That first dose to begin the study and randomization will be taken on site, and all of the first doses after that will be taken on site.
Okay.
Any other questions? If not, I will move on to the lab schedule, which is even easier than the visit schedule. So labs will be taken at 4 visits. Visit 1, the screening visit. Visit 2, the baseline visit. Visit 5, which is 3 months in. And visit 8, which is the termination visit, the 6-month visit. You have a pretty easy schedule of lab draws here. And you will also get a quick reference chart to tell you how to package them and send them off. Some of them are ambient, some of them are 4 degrees. And there's the biomarkers is really the only one that's at minus 20 that will be shipped out only 3 days out of the week, so you can store those and then ship those out together. And really, there's nothing like I said, very that you should have too many questions on this particular lab schedule. The draws are pretty simple. You're not taking too much blood from the subject, so you shouldn't have any issues with that. But I'd be more than happy to take any questions on this, and obviously you will get a copy of this as well as the lab manual and the distribution schedule for the different vials from MedPace, and we hopefully will get that out before the end of the year, all of those kits. Any questions? All right. Well, I will hand it over to Barbara. EDC. Yes, so I will be sending you an email in the coming weeks for your training for the EDC system. We will be using BioClinica for both the ECRF, which is called Express, and for the IRT system, which is called Trident. Those of you who have already used BioClinica before, you will only have to take a small refresher course for these systems. For those of you who have not taken them, it takes between 20 and 40 minutes for you to take the online training and then take the little test. It's not too difficult, so I don't anticipate any issues, but BioClinica is there to help if you do have any issues. And I am here to answer any other questions that for some reason BioClinica cannot answer. So feel free to go ahead and take those trainings as soon as I send that out. I believe I'll send out the instructions for the Express system in this week. For the IRT Trident system, that training won't be ready for another 2 weeks. And then you'll have to take that one as well. For those of you that don't know, the IRT Trident system is where you will receive drug and randomized patients, and the EDC system, Express, the ECRF, is where you will enter all the information for those particular visits. Any questions on that?
Will you be sending any screenshots so we can generate source?
Yes, so actually our monitors are actually here with us, and they will be generating the source documents for you. I will be sending the screenshots as well, in case you want to use those to help you. But the monitors will be creating source documents for you that they will bring to you on your SIVs. And some of them, if you ask for them, I'm sure they'd be happy to provide them beforehand.
Thank you. I think I just heard you're generating our source. Thank you.
Well, that's the least we could do after this AV scuffle, you know. But we do, as Omar said, we do have the monitors from Clinipace here. I think they're a great team. Some of them have worked with your sites in the past, and they gave you glowing reviews, which was a nice validation in addition to Omar's good, thorough site visits. So any other questions that you have with regard to the protocol or for NuSirt or for the monitors that we could answer at this time? I'll take that as a no. You really did miss a great introduction I made for our KOLs and for the NuSirt team, so I'll just have to somehow convey this under another medium at a future date. But again, we apologize for the snafu. It's probably crazy us to try and do it the Monday after Thanksgiving, but thank you for your patience with that. And we will have this recorded. It will be on the website if there are questions. Obviously, clearly from this, the monitors will be spending time going over the waist circumference and the blood pressure measurement, as well as any questions you may have when you have your SIV. And I guess with that, if there's no further comments or questions, I will wish you all a very good afternoon and hope you had a great Thanksgiving holiday and look forward to working with you.